Hello everybody, in this video I'm going to be talking to you about antibodies. So I'm going to be talking to you about the structure of antibodies and the function of antibodies. So you have just learned about the fact that antibodies are produced by plasma B cells. So B cells are floating about and then they come into contact with a foreign antigen, they become activated. When they're activated, they undergo clonal selections, they do rapid mitosis, and they differentiate into plasma B cells and memory B cells. Those plasma B cells differentiate, so they become adapted to produce antibodies, and antibodies are made of protein. So as we might expect, plasma B cells, they grow a bit and they develop lots of rough endoplasmic reticulum, ribosomes, mitochondria, and Golgi apparatus, because all of these things are important to enable them to do protein synthesis. So plasma B cells produce and secrete antibodies, and antibodies are quaternary proteins. They're made up of 4 polypeptide chains which are joined together by disulfide bridges, and we're going to have a look now at a diagram of an antibody and have a look at the different parts and the function of each of the different parts. So if we take a look at this diagram of the antibody, we can see that it's made up of 4 polypeptide chains joined together by disulfide bridges. And we can also see that you've got 2 long chains and 2 short chains. All 4 of these chains have a constant region, which is in green. This is just the same on every single antibody. Every single antibody that you produce in your whole life will all look exactly the same. What's amazing about antibodies is this red section that you can see. This is called the variable region. It's sort of a strange quirk in your DNA that enables you to produce many innumerable different shaped variable regions that just sit on the ends of your antibodies and essentially create all these different shapes that are potentially complementary to these myriad of different antigens that your body might be exposed to during your lifetime. So the fact that you can produce all of these different shaped variable regions is what enables you to produce complementary antibodies to these unique antigens that you might be infected with. So it's really the variable region that matters. And you can see here that the variable region is the site for attachment to the antigen. Can you also see that you've got a hinge region? This means that these arms can be closer together or they can be further apart, because each of these arms will bind onto 2 separate antigens, and maybe those antigens are close together on the surface of a pathogen, maybe they are far apart on the surface of a pathogen, or maybe they are on 2 separate pathogens. So these arms can move, they can be closer together or further apart, so they can bind onto different antigens in different places on the surface of different pathogens. So this hinge region is also important. And if we look at the other side of our antibody, we will see that we have got the phagocyte attachment site. This is part of the constant region. So every single antibody in your body has this phagocyte attachment site that is complementary to receptors on the surface of phagocytes, because these antibodies are going to eventually be absorbed by phagocytes and digested alongside the pathogens that they have grabbed hold of. And we'll talk a little bit in a minute about how they actually function. But just make sure you understand the structure and the importance of the different parts of the antibody. So I know I've just really drawn it as squares, but it's important that you can use your imagination a little bit and you understand that actually this variable region will have complex shape, and that shape, whatever it might be, will enable them to be complementary to a particular antigen on the surface of a pathogen. So what is the actual function of antibodies? This is where I'm going to introduce you to 4 quite long words. So there are 4 main functions of antibodies. There's no particular order of importance in terms of their function, so we'll talk about them in any particular order, but just so that you are aware, from now on we will be drawing antibodies like this, just for simplicity. Okay, so what can they do? Well, the first thing that they do is agglutination. Agglutination is when a single antibody will bind multiple antigens on different pathogens, causing them to clump together. Let's just have a think about that for a minute. So imagine we have got some pathogens, and they've all got antigens on their surface. The antibody can bind a number of pathogens together. So, do you remember we said they had those two variable regions? Just going to draw this quite simplistically here, but hopefully you can imagine what I mean. And they all get clumped together. This process is known as agglutination. This means that when these pathogens get phagocytosed, so once they get engulfed by a phagocyte, the phagocyte is not engulfing one pathogen at the time. It can engulf many tens or hundreds of pathogens all at the same time for phagocytosis to be engulfed and then digested and broken down. The second function of an antibody is immobilization. So imagine a pathogen that has a flagellum, and we know a flagellum is like a little tail that enables it to move around. If the antibody binds onto antigens on the surface of the flagellum, that flagellum is no longer able to move, and the pathogen cannot swim, it cannot move through your body, it cannot get into and invade tissues, and it can no longer cause harm. So it can't get to where it's going to cause harm, and not just that, because it can't move, it also cannot escape from the phagocytes, and so it makes it easier for the phagocyte to ingest. The third function of antibodies is called opsonization. Opsonization is just when an antibody binds onto the antigen on the surface of the pathogen, and the phagocyte attachment site enables the phagocyte to bind on readily because the phagocytes recognize the constant region, and it makes it easier for the phagocyte to ingest the pathogen. And the fourth function of antibodies is called neutralization. So we know that some pathogens produce toxins, for instance the botulinum toxin or the cholera toxin. These toxins can cause harm when they enter or bind with cells. So an antibody can bind to a toxin, so in this case the toxin is the antigen, and therefore block its access to cells, and so the toxin can no longer cause harm. When this is the case, the antibody is known as an antitoxin. Okay, so that is the basics of what you need to know about antibodies. What do you need to know about them? You need to know where they're made in plasma B cells. You need to know their structure, so the fact that they have 4 polypeptide chains, that they have this hinge region with the disulfide bridges, that they have the binding site for the phagocyte, and that they have the binding site for the antigen. And then you need to know about their 4 functions, so that opsonization, agglutination, immobilization, and neutralization. I apologise for my very simplistic drawing of the antibodies here, but I really would have been here all day if I'd done the little variable shapes on the end to demonstrate how their binding sites are complementary because they have this specific tertiary structure, but I'm just relying on you to understand that. And remember, being able to talk about this specific tertiary structure and the fact that it forms these complementary binding sites is going to be important when you answer your exam questions on this topic. So I'd just like to take a minute before we finish to talk to you about the relationship between antibodies and antigens. I think sometimes when we talk about antigens, we can be quite simplistic in our language, or we can just think about them as these very simple structures, you know, like on the diagram I'm just drawing them as little triangles, but you really need to be aware that this is just for the ease of our understanding. An antigen is a complex structure with a complex tertiary shape. So if we were to think about an antigen, so let's just take a minute to do that. So let's just imagine that this was an antigen on the surface of a pathogen. So we know an antigen is a molecule, usually protein-based, that can trigger an immune response. So we can see here, this antigen has complex tertiary structure. And when we think about an antibody, if we just go back to the antibodies, we talk about the antibodies having these binding sites that are capable of binding onto an antigen. Now, that doesn't mean that they bind onto the whole antigen. So if we just go back to our antigen here, the antibody might bind on to this particular part of the antigen. That could be the variable region, for instance, on our antigen. Sorry, on our antibody. So, this antibody has bound onto this part of the antigen. So, one particular B cell, so we've made a B plasma cell here, and this particular B plasma cell is able to make only one type of antibody. Let's just say, for simplicity, this one. But that doesn't mean that there can't be another kind of B plasma cell that's also triggered to be produced through clonal selection and expansion and mitosis. And this B plasma cell might produce a different antibody that has a different variable region that's capable of binding onto this area of the antigen. So when you have an immune response to something, so when you're exposed to a pathogen and that pathogen contains antigens, the first key thing to understand is that antigen has complex tertiary structure, and you can make more than one type of antibody against that particular antigen. So you can make antibodies that bind onto different parts of the antigen, and therefore you can have a range of different B plasma cells that produce those different antibodies. This is called a polyclonal response. So we have a polyclonal response. We are making lots of many different types of B plasma cells to produce lots of many different types of antibodies. In addition, if we just go back to thinking about our pathogen, so say this is our pathogen with its antigens on its surface, it doesn't just have one type of antigen on its surface. So obviously it's got this one that we've drawn here, but let's just take a minute to think about it. So let's imagine that this is our pathogen. It can have many different types of antigens on its surface. So you've got antigen 1, 2, and 3. And not only can you make different antibodies against these different antigens, but you can make different antibodies to bind to different parts of each one of these antigens. So it's not quite as simplistic as you might have been imagining it. So it's really important that you understand that when we have our normal immune response, we have a polyclonal response. So production of B cells with several different specificities occurs during normal infection. These are known as polyclonal antibodies. And in our body, we would expect to find really high concentrations of these polyclonal antibodies in our spleen, because we have really high levels of lymphocytes in our spleen. The next thing you're going to learn about in the next video is going to be about the production of monoclonal antibodies and how we can use monoclonal antibodies in diagnostic testing. So that's why this word polyclonal is important, because you need to understand the difference between polyclonal and monoclonal antibodies. Okay, so that's important because you might get asked questions about different tertiary structures of antibodies and why you might be able to have more than one type of antibody against the same pathogen. And that's the concept that those questions are really asking you to think about. So I think that's pretty much everything that you need to know about antibodies. So the structure, their function, and then this idea of diversity, so that there are millions of different types of B cells that exist in very small numbers in your body, and the ones that create the complementary antibodies are selected, and then they undergo rapid mitosis and differentiate to become plasma B cells that produce the antibodies and memory cells. The second really important concept is specificity. So the idea that only one specific plasma B cell is going to produce the particular antibody, and antibodies have complex tertiary structures that are complementary to only one antigen. And this idea of binding and complementary shapes is just one that you find all the time in biology. Okay, so we're going to leave it there for now. Make sure that you learn all of the material we've just talked about, and the next thing you're going to learn about is monoclonal antibodies. Well done.